Good morning, everyone. I'm John Christian, your reporter for today. We are going to tackle about genetic code.
Before we proceed to our main discussion, first let us know the terms that we will going to encounter throughout our lesson. First, the DNA or the deoxyribonucleic acid. It is the hereditary material in humans and almost all other organisms. Nearly every cell in the person's body has the same DNA. Next is the RNA or the ribonucleic acid. It is a molecule similar to DNA. Unlike DNA, RNA is a single strand. An RNA strand has a backbone made of alternating sugar or ribose and a phosphate groups. The next one is the codon. It is a trinucleotide sequence of DNA or RNA that corresponds to a specific amino acid. Anticodon. It is a trinucleotide sequence complementary to that of a corresponding codon in a messenger RNA or the mRNA sequence. Transcription. It is the process by which the information in a strand of a DNA is copied into a new molecule of messenger RNA or the mRNA. Translation. The process of translating the sequence of a messenger RNA or the mRNA molecule to a sequence of amino acids during protein synthesis. Amino acids. It is the molecules that combine to form proteins. Genetic code. Genetic code is the term we use for the way that the four bases of DNA, the A, C, G, and T, or the adenine, cytosine, guanine, and thymine, are strung together in a way that the cellular machinery, the ribosome, can read them and turn them into a protein. So we know that the DNA provides instruction for making proteins. And we have this sequence of bases, the A, C, G, and T, which determines our unique genetic code and provide instructions for producing molecules in the body. And in a DNA, the cell reads the code in groups of three bases. And this group of three or each triplet of bases will be called codon, which specifies for an amino acid. And this codon will be used in synthesizing protein. Characteristics of genetic code. The genetic code has a number of important characteristics. First one, the genetic code is universal. All known living organisms use the same genetic code. This shows that all organisms share a common evolutionary history. We know that all organisms use DNA to store genetic information. They all use the same four bases, which are the guanine, adenine, cytosine, and thymine, and they make the same amino acids. This is why DNA provides us with a universal genetic code. Because of the genetic code, a lot of very different species have the same genes. The similarities and differences between the same gene in two different species can tell us how closely related they are. Second one, the genetic code is unambiguous. Each codon codes for just one amino acid. So when we say ambiguous means more than one, but non-ambiguous means only one. So for example, the codon CCU, it only encodes for proline. The AUG, it only encodes for methionine or met or the start codon. The third one, the genetic code is redundant. Most amino acids are encoded by more than one codon. A codon is usually a group of three nucleotides that encodes for a specific amino acid. Every codon is generally specific for only one amino acid. For example, both AAG and AAA codons code for the lysine amino acid. The lysine produced by AAG shows similar functions to the lysine made by AAA. The silent substitutions occur because of the redundancy of the genetic code. How does genetic code works? First, it undergoes transcription. It is the transfer of genetic instructions in DNA to mRNA. So, transcription, it is a process in which information is rewritten, or it is the process of copying out the DNA sequence of a gene in that similar alphabet of RNA. It uses DNA as a template to make an RNA or mRNA molecule. During transcription, a strand of mRNA is made that is complementary to a strand of DNA. Eventually, portions of the transcribed mRNA will be made into functional proteins. After transcription, translation follows. So translation, it is the process in which the genetic code in mRNA is read to make a protein. And it occurs in the cytoplasm following DNA transcription, and like transcription, it has three stages: initiation, elongation, and termination. During translation, mRNA nucleotide bases are read as codons as of three bases. Each codon codes for a particular amino acid. Every tRNA molecule possesses an anticodon that is complementary to the mRNA codon, and at the opposite end lies the attached amino acid. tRNA molecules are therefore responsible for bringing amino acids to the ribosome in correct order, ready for polypeptide assembly. Codons. Cells decode mRNAs by reading their nucleotides in groups of three called codons. Codons in an mRNA are read during translation, beginning with a start codon and continuing until a stop codon is reached. So for example, on the picture, you can consider a codon if you can see a sequence of three nucleotides that corresponds to specific amino acids. So in the picture, you see AUG, where it consists of adenine, uracil, and guanine. So there's three sequence of bases. So it is a group. So it is called codon. So that codon AUG corresponds to amino acid methionine, or a start codon. Genetic code table. The full set of relationships between codons and amino acids is called genetic code. By reading a genetic code table, you can determine the amino acids that will be brought in by the tRNAs. You can determine the sequence of amino acids that make up a protein. And if you're wondering why this matters, understanding how amino acids are coded for help us understand the human traits and how changes in amino acids have the potential to alter those traits, not just for humans, but all living organisms. The codons and their corresponding amino acids have been discovered by scientists. There are many ways you can represent the information in a chart. The most common ways are the rectangular codon chart and the circular codon chart. Reading the genetic code. The start codon establishes the reading frame of mRNA. The reading frame is the way the letters are divided into codons. After the AUG start codon, the next three letters are read as the second codon. The next three letters after that are read as the third codon, and so on. This is illustrated in the figure. The mRNA molecule is read codon by codon until a stop codon is reached. UAG codon, UGA codon, and UAA codon are all stop codons. They do not code for any amino acids. Stop codons are also known as termination codons. I'm going to present a video for you to see on how the genetic code table is used in translation or translating mRNA.
Here we have an mRNA molecule. To translate it into a polypeptide, I'll scan for the start codon, then I'll consult my chart for all the remaining codons until I reach the stop codon. So I scan, I find the start codon AUG, which, if I consult my chart, codes for the amino acid MET, or methionine. I then move to the next codon, CCG, which codes for the amino acid PRO, which is proline. Then we have CGA, coding for arginine, and GUG, coding for valine. Then UCG, coding for serine. And then finally we hit UGA, which does not code for an amino acid. It's simply a stop codon signaling the end of translation. Any codon after the stop codon does not get translated. At this point, I've translated the full mRNA molecules into a sequence of amino acids that will make up a polypeptide that will then be modified and folded into a protein to use in the cell. In reality, the mRNA molecule will be much longer than this, with many more codons, resulting in a polypeptide chain that contains many more amino acids. For example, the enzyme Rubisco, which is used in photosynthesis, is made up of around 500 amino acids.
The summary of this lesson. The genetic code consists of the sequence of bases in DNA or RNA. Groups of 3 bases form codons and each codon stands for one amino acid. The codons are read in sequence following the start codon until a stop codon is reached. The genetic code is universal, unambiguous, and redundant. Thank you for listening.